Phases of growth. The period of growth is generally divided into three phases, namely first phase meristematic, second phase elongation, and third phase is the maturation phase. Now we study it by looking at the root tips. Now the constantly dividing cells both at the root apex and shoot apex represent the meristematic phase of growth. Now what are the characteristics of cells in this meristematic phase of growth? The cells in this region are rich in protoplasm because they needed to divide, so they have rich in protoplasm. They possess large conspicuous nuclei that we can see it, large conspicuous nuclei, and their cell walls are primary in nature. Of course it will be primary because they will be going to divide again and again, so their cell walls remains primary in nature. Thin, of course it needed to divide, so it's thin and cellulosic with abundant plasmodesmatal connections. Okay, so they are rich in protoplasm, they possess a large conspicuous nuclei, their walls are primary in nature, thin and cellulosic with abundant plasmodesmatal connections. Now the cells proximal, just next away from the tip to the meristematic zone, represent the phase of elongation. So after the meristematic zone is the phase of elongation. Okay, just proximal. Now increase vacuolation, that the vacuolation is kind of a space in the plant cell. Now cell enlargement, that has the water. Now cell enlargement and new cell wall deposition are the characteristics of the cells in this phase, right? New cell wall deposition. So in the meristematic zone it was primary. Now it's a new cell wall deposition. Now further away from the apex, that is more proximal to the phase of elongation, lies the portion of axis which is undergoing the phase of maturation. Now the cells of the zone attain their maximal size in terms of wall thickening, protoplasmic modifications. Okay, so this was it. Detection of zones of elongation by the parallel line technique. Zones ABCD immediately behind the apex have elongated most. So ABCD has elongated most.